champions, I welcome you all to score 360 out of 360 in NEET Bio within 250 days, and today we are going to talk about linkage, recombination and chromosomal theory. And I aptly remember when I was in school, this topic was too troublesome for me. I was not able to understand initially, but then gladly I got a good teacher who explained it to me really well, and I'll try my best to explain it to you in the same way. Okay, so over here, let's go ahead children, let's see what do we have in hand. What did we study in the previous session? We spoke about inheritance of two genes, that is, we spoke about dihybrid cross. So until now we were just talking about monohybrid cross, but yesterday we spoke about dihybrid cross. We spoke about the ratio, phenotypic ratio, genotypic ratio, which is very much important. Okay, so just go through that if you have not studied. And also one more thing, if you want the notes PDF, then you can join our WhatsApp channel, which will include just us. Now the number of questions that came the previous year was 6. Now 6 into 4, 24 marks in total. So as I said, even in a shorts that I made, this chapter and molecular basis of inheritance holds a very important part in the NEET exam. So you cannot miss this chapter at all. And I also remember I gave you homework. Let's see who all got it correct. In a cross between capital A, capital A, capital B and small a, okay, I'm just writing, and small a, small b, the ratio of F2 genotypes between AA, AB, okay. So basically this is exactly like the dihybrid cross that we studied, where instead of this it was round, yellow and wrinkled and what children? Green. Same like that, okay. So when you fuse it, when you fuse all of them, you get one thing. From here there is A gamete, here there is B. So here it is A and then again B. So basically all of them is AA, BB. We studied about it earlier, okay. For example, from this only one chromosome can come, from this only one chromosome can come. So basically a gamete will be having either AB, whatever you do it will be just AB in one particular gamete. Here also it will be just A and B in another gamete. Together if you fuse them you get something like this. Now if you do self-pollination, okay, now when you do self-pollination you will get the F2 generation. Okay, so when you cross this again with this, you will get the F2 generation. But over here, instead of asking about the F2 generations ratio, they have mentioned about the genotypes. They are not asking you about the phenotypic ratio of 9:3:3:1. That is not the question. This is the phenotypic ratio, as in how do they look. They are asking you about the genotype, and they have mentioned the genotype as well. So over here, it is capital A, capital B, capital B. So we have studied in genotypic ratio, the ratio goes like this: 1:2:1, 2:4:2, and 1:2:1 again. So 1:2:1, 1:2:1, in middle you double it. Okay, here the one starts with the dominant parent, which is this, so it will be AA, BB. Okay, another one ends with the recessive parent, which is this, so it will be AA, BB. So you got two things. This is 1, you got this information that this is 1 and this is also 1. In between both of them will be 2 and 2. Here if you see 1 and 1 also, you will get the answer. No one starts with 1 except this and ends with 1, right? And both of them which comes in middle, they are 2 and 2, the 2 and the 2. Okay, so please, if you do not know the genotypic ratio, as of now I'm not making a whole Punnett square and explaining you each and every genotype. There will be much more genotypes than this, because as I said, for each genotype you have one. So there will be one parent, one F2 progeny which will be having this genotype, which is why I wrote 1 over here. There will be one F2 progeny which will be having a genotype like this, which is why I wrote 1 over here. Similarly, you have so many, you have to find out using the Punnett square. Okay, try it out once and then it will be sorted. Okay, now going ahead children, okay, let's start with today's topic. And when you start with today's topic, you have to know that what was Mendel's role in this. So you all know that Mendel was a person who was working on pea plants, the garden pea plant, right? Garden, please remember it. And his work went up for 7 years. From 1856 it went up to 1863. 7 long years, you have to remember the years as well. Now his work, even though it ended up in 1863, it got published in 1865. First very important information is that what was the date of publishing of Mendel's work? Okay, so I am writing Mendel's work got published when children? published 1865. But you know what the tragedy was? Until 1900, this work was not recognized. Until 1900, that is so many years, like 40 years, something like that, 35 years. Until 35 years, it was not recognized. Only in 1900, it was starting to get recognition. But what were the loopholes? Why didn't it get the enough recognition that it needed? So there are a few things that is exactly written over here. We'll study like this. First thing, communication was not easy. As you know, in 1865, you cannot expect communication to be easy. The mobile phones, everything has to come in. Everyone has to get this, then only they can communicate easily. So one point, that is the first point, is communication was not easy. So I'm just writing it. Okay, the loopholes. The problem was not with his work initially, it was because of communication. Okay, so I'm writing lack of communication. Fine. Second thing, his concepts of genes, he didn't know genes. According to him, it was factors. Okay, are stable and discrete units that control the expression of traits and of the pair of alleles which did not blend with each other was not accepted by his contemporaries. As in, they are trying to say that he said that, okay, there is a chromosome, there is another chromosome, this stands for tall, this stands for short. When a plant has both of this, one dominates over the other and it will be tall. Okay, and then if a chromosome has small t, the other chromosome has small t, then it will be dwarf. But he never mentioned about the middle heighted things. What about the plants which had middle heights? What about us who have different type of heights? Okay, who have different variation in a character, that is height. You never know about it. So his concept of basically the chromosomes are not blending with each other, or the genes are just there on one chromosome and it's not getting like there is no blending happening, there is no crossover happening, that was not actually accepted because people saw variety of characters around there, variety of traits in a character. Okay, so for example, height is a character, then tall is a trait and dwarf is a trait. Similarly, you have middle heighted things also. This was not explained by him. Okay, so I will write his concept of gene as a discrete unit was not accepted. Okay, it was not accepted. Now going ahead, thirdly, Mendel's approach for using mathematics to explain biological phenomena was totally new. So you also know, if I start initially, if there is no one around who has too much amount of information at this point of time, someone is working on biological things, along with he is using statistical approach, he is using mathematical approach, obviously others would also not be understanding. You never know, their wavelength would be obviously different. Okay, they might not be able to understand this particular use because it was not very common at that point of time. So mathematical use was not what do I say, acceptable. Okay, it was not acceptable. So communication, okay, his concept of gene as a discrete unit which does not show blending, that, and use of mathematical applications. Now next thing, finally the Mendel's work suggested that factors were discrete units. He could not provide any physical proof. He was just saying that, okay, I cross a tall plant, I cross a dwarf plant, I get all the plants as tall. But he was not able to show, and he was saying that there are factors responsible for this, but he did not show this. There was no physical proof. Why there was no physical proof, children? Because at that point of time they did not have this thing, what do you say, microscope. Okay, or even if it was there, people Janssen and Janssen, the people who were there responsible for the finding of microscope and everything, for them to communicate that we have something, that also needs a source of communication, right? That was also not possible, which is why there were certain problems almost for 35 years. 35 years, you just imagine you being 35, that many years, for that many years his work was not recognized. Mendel's work was published in 1865, please remember it. Until 1900 it did not get any recognition. Then now going ahead, rediscovery. Now this is a time when Mendel's things came back. So in 1900, 3 scientists, name were De Vries, Correns and von Schermach. Okay, please remember it is De Vries, Correns and von Schermach. 3 people, 1, 2, 3. They were the ones who rediscovered the work of Mendel. Discovered the work of Mendel. Okay? And I told you in which year it was rediscovered. It was rediscovered in the year 1900, because at this point of time they got the microscope and everything, so they were able to see a lot of things. So they independently, okay, they did not come together and discover it, independently rediscovers Mendel's results on inheritance of character. Also by this time, due to advancement in microscopy that were taking place, scientists were able to carefully observe what? Cell division. Okay, this led to discovery of structures in the nucleus that appear to be double and divide just before each cell division. These were called as chromosomes. So at this point of time, when microscope and everything was developed, it came to notice that, okay, if there is a nucleus, then inside the nucleus there are something like this which are known as chromosomes, which are going to divide itself during each cell division. Okay, which are going to be apart from each other during each cell division. Okay, by 1902, chromosome movement during meiosis had been worked out. So now comes the era of Sutton and Boveri. So there were 2 other scientists, their name was Sutton and Boveri, and their story happened in the year 1902. Please remember that. Okay. Now these people were able to see that chromosomes were moving during cell division, especially when they were talking about meiosis. Meiosis happens during gamete formation. Okay. Now Walter Sutton was his name. Okay, one person's name was Walter Sutton and other person's name was Theodore Boveri. Guys, these are important information, MCQs have come. Okay. Now noted that behavior of chromosomes were parallel to the behavior of genes and used chromosome movement to explain Mendel's law. So Mendel always used to say that, for example, if some plant is tall and the other is dwarf, basically if you have 2 factors, these factors are coming down and now you have a pair of factors. So similarly, they also went around, okay, they were saying that, okay, if he is saying that something is coming together and forming like this, so these are factors that Mendel was talking about. These guys, they found that, okay, probably then these factors might be on the chromosome. These factors might be on the chromosome and these chromosomes might be coming together in an organism. So basically Mendel's work, according to him, there were factors. He did not have any physical proof and he was saying in a pair of factors, one we were getting from both of our parents. Similarly over here, probably there would have been a pair of factors, and from this pair you get one factor down, right, according to Mendel. Similarly over here, in a pair of chromosome that you see, one pair, another pair, one chromosome comes down and you have a new pair. Okay, so this was actually linked by Sutton and Boveri. Their name was Walter Sutton and Theodore Boveri in the year 1902. They found that probably the Mendel's thing that he was saying that factors move down, it would be with the help of chromosome. Probably chromosomes would be the one that would have these factors on them. Okay, so recall that you have studied the behavior of chromosomes during mitosis. What do you mean by mitosis, children? Equational division, and during meiosis, reduction division. The important things to remember are chromosomes as well as genes occur in pairs. The 2 alleles of a gene pair are located on homologous site of the chromosome. What do you mean by that? Same thing that I explained over here. Homologous chromosome as in, there is a chromosome like this, okay, and it has one factor according to Mendel. There will be another factor of the same trait over here, same height, same character, I mean. Over here, it can be capital T or it can be small t. So, same thing they are saying. These chromosomes which have a pair of gene, okay, a pair of allele, sorry, a pair of allele. This is an allele, this is an allele for the gene coding for height, right? So these alleles are there on homologous chromosomes. Now even still if you are not able to understand, so this is a pair of chromosome, this is another pair of chromosome. Now there is R and small r. I cannot say that this chromosome is homologous to this chromosome. The homologous pair of this chromosome is this one because it has the opposite allele for the same character, whereas the homologous chromosome for this one will be this. Okay, that is what you mean by homologous chromosome. So I hope you got this. Now in your NCERT there is a picture also that is given over here. Ayo, wait, where is that picture? Didn't I put it? Okay, I didn't put it. Okay, so that was the thing, right? Now according to Sutton and Bovary, they wanted to put down certain things there. Okay, so they put down their findings and named it chromosomal theory of inheritance. So until now no one was aware of that, but now people got a clarity about it, and Sutton and Bovary were the one who said that pairing and separation of a pair of chromosomes would lead to segregation of a pair of factors they carried. So pairing and segregation of a pair of chromosomes would lead to segregation of the pair of factors. Now this will also be segregated. That was what they said. So a pair of chromosomes, segregation of a pair of chromosome would obviously lead to segregation of a pair of character, okay, a pair of factors that they were carrying. Simple. Segregation of a chromosome would segregate the pair of factors because obviously chromosome has the factors. This is what they said. Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it chromosomal theory of inheritance. Actually, both of them were responsible for chromosomal theory of inheritance, but in the NCERT last part it is said that Sutton was the person who united the knowledge of Mendelian principles and his things, and then they came out with chromosomal theory of inheritance. Fine, that is done. Now, there were certain roadblocks in this. Okay, there were certain roadblocks in this, and I told you previously when we were talking about law of independent assortment. According to law of independent assortment, what happens, children? Basically, the chromosomes, they segregate independently. For example, I have two chromosomes for two characters. Over here, just see this. Over here, just see this. So this is a pair of chromosome for one character. This is another pair of trait. This is another pair of character. This is another pair of character. Now, when I want to put down into gametes, this will be assorting independently of this. For example, one will get green and red, other will also get green and red. Similarly, over here it gets orange and green, right? This is the normal case that we studied. So probably in this case we might think that it is not independent, it is dependent, because green is always with red, right? Green is always with red, whereas orange is always with dark green, right? Over here also you saw like that, but there is a different possibility as well. There's a different possibility as well, where what is happening, the green, if you see at the end over here, it is with dark green. So this has been shifted. This has been shifted. So over here dark green was here. Now this time the dark green chromosomes are over here and the red is over there. So this can happen. They're independent. This movement of this particular chromosome is independent of the movement of this particular chromosome. You cannot link them. This is the law of independent assortment. They gave a picture of this, as simple as that. Okay, so if it was not getting independently assorted, then probably always light green would have been with red. Always light green would have been with red. But over here you see light green is with dark green, because this chromosome and this chromosome are assorting independently. Okay, this is what was said. Okay, this is what was said. Actually, this particular picture should not be put here. I don't know why they put it over here. They should have been putting in law of independent assortment, but still, since it is given, we have to explain. So please do not get confused with this and this. Okay, not get confused with this and this. This is law of independent assortment that they are trying to show, but over here they are trying to say chromosomal theory of inheritance. In that they are saying whenever a pair of chromosomes gets segregated, that's when a pair of factors which are on them, they also get segregated. Okay, and now you can add that this is independent assortment. This also shows independent assortment. Basically, actually this does not show independent assortment, so I should not be taking that. But if there were two chromosomes, then definitely. If this was this chromosome and this chromosome, then definitely they are showing independent assortment. this and this can go, or this and this can go, either way. Okay, no bounds attached. I know I got you confused. Please put down all of your confusion away. Let's just concentrate on chromosomal theory of inheritance and also independent assortment. Both are different things. Okay, now going ahead, children, there is a box that is given over here, but we will be discussing this box later, okay, at the end of the class. Now, human beings basically do not believe on things. Can you believe on things randomly said by people? No, we cannot. That is our human nature. So basically what has to be done, we have to experiment on things, then only we can come to a conclusion whether this is actually true or not. So now, chromosomal theory of inheritance was in the verge of experimentation, and this experiment was done by Morgan. Please remember, okay, the verification. Morgan was the one who did the experimental verification of what, children? Chromosomal theory of inheritance. Okay, and for this he didn't use the garden pea plants. He said there's too much work on garden pea plants done already. I want to check this on something else, and this something is tiny fruit flies, which you call as Drosophila melanogaster. So there are fruit flies, you call them, what the scientific name is, Drosophila melanogaster. Now there are few key terms that is given over here, that why did he actually chose Drosophila melanogaster. First thing, they were grown on simple synthetic medium, so growing them was very easy. Growing in lab was easy, agree? Next thing, they complete their life cycle within about 2 weeks. So lifespan is just 2 weeks. So I just have to wait for 2 weeks to see my results. That's all. If I am a scientist, I just have to wait for 2 weeks to see my result. And a single mating could probably produce large number of offsprings. So I can study on so many offsprings. So large number of offsprings. Okay. Now what? There was a clear differentiation between the sexes. So male and female had clear differences. Okay. Next thing, it also has many type of hereditary variations. So there were so many variations that could be observed even on low power microscope. So I am writing that they holded more variations than you can imagine. Okay. So why growing them in lab was easy? The lifespan was just 2 weeks. There was larger number of offsprings. The male and the female sexes could be easily differentiated. So I can help it would be helping in fusion, right? I can do the fertilization over there with the help of this. Now more variations were also found. More variations means I can find more amount of differences in them as well, more amount of study as well, right? So Morgan was the one who did experimental verification for chromosomal theory of inheritance. But while he was doing this, okay, and he was doing on Drosophila melanogaster. Please remember it. He was doing on Drosophila melanogaster. Okay. Now, while he was doing all this thing, he termed out two major things, which is, children, linkage and recombination. And this gave the answer for a lot of things, that why there are different heights, that why there are so many different characters apart from the things that is being mentioned by Mendel. How can different characters come in without blending? So basically all these things were explained with the help of these key concepts, which is known as linkage and recombination. Now for you to understand this, I will make it like this. And this was the exception to independent assortment. Why it is an exception to independent assortment, I'll tell you. So basically, if I have a chromosome like this, I have another chromosome like this, and I have two more chromosomes like this. Okay. Now, according to law of independent assortment, there is one pair of factor over here, okay, and another pair of factors over here. So for avoiding confusion, I'm just writing Acha, wait. Okay, small r. Okay. So one pair of character is on one chromosome, another pair of characters on another chromosome, and now when the chromosomes are segregate, one among them will be going, so either it can be capital R or it can be capital Y, right, or it can be, so instead of writing like this, I will write like this. So the combinations of gametes that I can find, in a gamete from one pair one can come, so basically R can come, here Y can come, right. Similarly, next combination is with R, small y can come. Okay, now with small r, big Y can come, with small r, small y can come. So these were the possible gametes over there. So it was, Mendel said that they were all independently assorting, that it does not, it didn't matter that capital R has to always go with capital Y. It was not like that. It could go with this or this. This was independent assortment. Okay, they, for one pair of character and for another pair of character, the genes were independently assorting. That was the thing. But there is a situation. There is a situation, that is, instead of having two chromosomes, what if on one chromosome itself you have both the genes? Then what will happen? If on one chromosome itself you have both the genes, then is this possible? No, because if I give this chromosome, then always it will be RY. If I give this chromosome, then always it will be small r, small y. Agree? Do you agree with this? The gametes will be just having this thing. This is not possible then. If the genes for different characters are on the same chromosome, then can you expect it to be different? No. This is what you mean by linkage. Basically, linkage is there to describe the physical associations of genes on a chromosome. So I say if these gene pairs, basically two of them, round and yellow, if they are very close, I say that they are closely linked to each other. Closely linked to each other. But on the other hand, if this situation was like this, where the gene color, that is yellow, was located somewhere here on the same chromosome, but it is somewhere there. So I say it is loosely linked. It is loosely linked. It is not closely linked, it is loosely linked. Okay. Now, what is the use of loose linkage? What is the use of loose linkage? If they are tightly linked, then obviously, always the gametes will be like this. But if they are loosely linked, here comes the role of the next thing, that is known as recombination, which is responsible for a lot of variations in us. So what happens in recombination is that the chromosomes, while they divide, okay, the chromosomes while they divide, why did I do that? I don't know. It accidentally happened, children. Please forgive me. Okay. Now, the situation is where capital R is here, capital Y is here, small y is there, and small r is here. These are loosely linked. Okay. Now, in this situation, in our body, the chromosomes basically do crossover during cell division. What do you mean by crossover? Basically, they come together like this. Okay, the pairs, they basically come together like this, and they kind of exchange a lot of things between themselves. Okay, they exchange a lot of things between themselves such that now, if I shaded it in green and I shaded this in red, okay, and now these particular portions got exchanged, then the new chromosome will be having something like this. So it is R over here, Y over, small r over here, because here recombination does not happen. Here recombination did not happen, which is why both of them are same. But over here, this piece got cut and came over here, this piece got cut and came over here. So basically, this will be small y now and this will be big Y now. You see? Did you see this? This is known as recombination, children. Recombination is a result of crossover. So basically crossing over of the chromosome, which is a very natural process during cell division. At this point of time, what will happen, children? There will be new recombinant DNAs that will be formed, or recombinant chromosomes that will be formed. Now the gametes, if you see, it is capital R, small y, small r, capital Y. So if recombination was not happening, in that situation what would have been the gamete, children? If recombination, this thing, just this thing, if it was not happening, then the gamete combination would have been what? This chromosome comes separately, this chromosome comes separately. So there is RY and then there is small r, small y. So don't you think 50% of chance is there for recombinant DNA to come, and there is 50% of chance for this particular thing to come? Yes, recombination has 50% chance, and normal things for the parental variety to come. This is parental variety. This was there in the parent. Now that to come also is 50%. Agree? Agree? This is a very, very, very important piece of information. Now it is when they are loosely linked. When they are loosely linked. But what is the other case where they are tightly linked? For example, you have the chromosome. Okay, now over here you have capital R, over here you have small r, over here you have capital Y, over here you have small y. Okay, at this point of time, whatever recombination you try to do, the recombination would not be possible. You can cut and paste, whatever you do, it's not very much possible because they are closely linked. They are closely linked. Okay, now you think when gamete formation occurs, still I will be getting capital R, capital Y, small r, small y. So with this, if I keep on crossing and I come to a conclusion that basically I am getting the parental varieties only, that means the genes are closely linked. The genes over here are closely linked, which is why the recombination is less. So with this, I got an inference. With this, I got an inference, that is more linkage. There is more linkage, there is less recombination. Okay, there is less linkage like this, so there is more recombination, children, more chances of them to divide. You can always think like this: if you are more close, okay, if you are more close to each other, then probably you understand things better, there will be less fight. So more close, less fight. Okay, on the other hand, less close, more fight. Can think like that. Okay, so basically over here the linkage as in when they are close. Okay, if it is loosely linked, that means they are far away, that means more amount of recombination would be possible, that means different type of gametes can be formed, whereas on the other hand this is also possible. So don't you think this is an exception of law of independent assortment? Because over here in according to law of independent assortment, a pair of character, gene for a pair of character, okay, a gene for a pair of character assorts independently for another pair of character, but it's not happening like that. Both of them are on the same chromosome, then how can they assort independently? They are not independently assorting. For example, if they are closely linked, they are coming out like just like that. If they are not closely linked, then obviously recombination is happening, but still it is not following what law of independent assortment in total. Okay, so this is an exception where you can find linkage and recombination. So certain while he was doing his studies on Drosophila melanogaster for things that he was working on, okay, he wanted to prove chromosomal theory of inheritance, but during this point of time he discovered linkage and recombination, which is very much important, and recombination is the one thing that gives lots of variations in us also, which is why we can see different heights, different type of non-parental combination as well. Okay, I hope this is actually being clear to you. If you are not able to understand things, guys, you can slow down, you can pause, just have a quick read and then come back. Okay, so over here Morgan and his group knew that genes were located on X chromosome. Okay, so basically when he were talking about Drosophila melanogaster, he was working on traits which were there on not the normal chromosomes but the sex chromosomes. And which sex chromosome, children? X chromosome. Okay, these genes that he was talking about were there on the X chromosome. We will be studying about that. And saw quickly that when two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combination were much higher than, okay, so when two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental gene combination were much higher than the non-parental type. So the proportion of parental gene combination was much higher than the non-parental type when both of them were located on the same chromosome. Now they didn't say about linkage yet, but they will tell. Now Morgan attributed this to the physical association or linkage between the two genes. That is linkage to describe the physical association of a gene on a chromosome. Of genes on a chromosome, and the term recombination to describe the generation of non-parental combination, that is as this. Okay. Now Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were tightly linked, which showed very low recombination, while others which were loosely linked as this showed higher recombination. Okay. For example, he found that genes white and yellow were tightly linked. We will be talking about white and yellow, which is what they have said over here, were tightly linked and showed 1.3% combination. As in, you just try to understand that probably there could be a gene over here, which is what? Basically, this is a chromosome, and they are saying there is a gene for white and there is a gene for what, children? Yellow. Okay. These were tightly linked, as in this was not able to show recombination, which is why all the gametes that would be probably produced, all the progenies would be produced, would be having a chance of having this, because they are tightly linked. You see, the gametes were always like this. The gametes were always like this because they were tightly linked. Whereas, and showed 1.3% of recombination. So recombination rate was really less since they were close together. So whatever mixing matching do, somehow you end up in this. Now, while white and miniature wing, so this is white and this is white and yellow. Okay, that might be the color of the wing and the color of the eye. There are two characters. Now there's a third character that they are talking about, that is about the size of the wing. So about the size of the wing, it's saying that they showed high amount of recombination. Why? Because white is over here, the wing size is over here. These links are not closely associated, which is why they can show recombination. They can show recombination. This part can go to some other chromosome and some other chromosome's part can come to this, which is why it was different from the parental combination. So parent probably had white eye and short wing, but now this things has changed. It does not have short wing because the short wing trait is basically not closely linked. It showed 37.2% recombination. So how far it is, that means more amount of recombination is possible. If you are not able to understand this, please don't worry, we have an example. We will be studying with an example properly. Okay. And then his student Alfred Sturtevant used the frequency of recombination between gene pairs on the same chromosome as a measure of distance between the genes and mapped their position on the chromosome. So he was a person, okay, please remember his name is Alfred Sturtevant, okay, Sturtevant, you can remember like that, Sturtevant, and he was responsible for gene mapping. He was responsible for saying that, okay, probably the linkage, if it is this much, then recombination would be more or less, things like that. Okay. So frequency, he said frequency of recombination between gene pairs on the same chromosome as a measure of distance between the genes mapped. So if they were showing too much amount of recombination, that means the genes would have been located in a different place. If there was too less amount of recombination, that means probably the genes were close with each other. Okay, so frequency of recombination between gene pairs on the same chromosome as a measure of distance between the genes was mapped. Okay, so gene mapping was done by Alfred Sturtevant. He was the student of Morgan. Okay, now as promised, there is a given example in your book which is very, I mean, in the picture it blows my mind. So over here I will make for you. He was working on Drosophila melanogaster, agreed. Okay, and he said the genes which he was talking about were located on sex chromosome. So females have 2 X chromosome while the male have 1 X chromosome and 1 Y chromosome. So in the Y chromosome I cannot find this particular gene that I am talking about. Okay, even though it's homologous, but still I cannot find it. So basically over here the characters that I took was, okay, where shall I write? Yellow and white. Yellow and wait. Yellow and brown. Okay, that was the body. Yellow and brown body color. Okay, now you talk about eye color. One is red and the other one is white. Okay, now you need to know the dominant trait and the recessive trait. This one is dominant, children. This side, brown and red are dominant. So there's a little twist, so that means these are recessive. So how can I represent this? I need alleles formation, right? So for yellow I show with small y, for brown I show with small y plus, Okay, so recessive trait you're taking as the first letter, that is something different, but it was done like that. Okay, so plus sign I am putting for dominant showing. Now similarly over here, white is a recessive trait, so I am taking W, and over here it will be W plus. Okay, so this is a pair of pair of gene, and this is another pair of gene. Cool. Now I am saying the female which was there, as you can see over here, it was yellow and white. So for it to be yellow and white, that means both the alleles, both the chromosomes should be having the this thing, recessive one. So small y, small w, small w. Okay, and I am saying the male was, so this is yellow white children, this is yellow and white. Okay, and I am saying the male was brown and had red eyes, both the dominant character, brown and red eyes, it was dominant character. So now in Y there is nothing, Y chromosome there is nothing, so I cannot, so basically this will be showing brown and red eye, the dominant character. Okay, and this type of animals I call them the wild type, which has the dominant traits. Okay, now let's form the gametes. Now if this gametes come or this gamete come, either the same thing will happen, same thing is going to happen, because both has the same genes, so Y and W. Okay, that is one thing that mothers can give. Over here father can give either this, where there is Y plus and there is W plus. Okay, it can give either this, or now the second combination that can happen is the mother, okay, the mother give this. Now second combination that can happen is mother is giving this again. Even if she gives this, the same thing happens. That is why I am not mentioning, for avoiding confusion. And now father is giving the Y chromosome. Once he gave this chromosome, once he gave this particular chromosome. At this point of time, you see, now this is X chromosome, agree? This is an X chromosome, this is also an X chromosome, so this is a female. And this female, now if you see, it has Y and W plus as dominant traits over here. So this female will be having brown and red eye. Okay, whereas if you talk about the male which has the Y chromosome and one X chromosome, this will be now yellow and white. Okay, this will be now yellow and white. So this twist has happened. So over here now female is wild. Over here now female is wild. Now if you want to fuse them for F2 generation, okay, now let's try to fuse them for F2 generation. So over here they can possibly give two type of gametes. Okay, where shall I write it? I shall write it here. Okay, listen to me very carefully. Okay, over here it can give two type of gametes. One can be small y, small w, that is this. Other can be capital Y, or basically Y plus and W plus. Okay, now next thing, this chromosome can come or this chromosome can come. If this is coming, then small y, small w, and if this is coming, then nothing is happening over there. Okay, now over here you see this and this combination happens. So small y, small w, small w, this will be yellow and white. Okay, now this and this, it will be Y plus Y and W plus W. Agree? W plus and W, this and this, right? So this will be what, children? Brown and red. Is this visible to you by any chance? Yes. Now this and this fuses, so obviously there is male gamete, so over here it has to be yellow and white. Okay, now over here if you try to do that, then it will be Y plus Y, W plus W. So again it will be brown and red. So if you see the possible genotypes that you can see over here, children, either is this or it is this. 25% of chance it could be this, 25% of chance could be this, if recombination is not happening. If recombination is not happening, things would be like this. Now suppose recombination has happened, so this is going to come here and this is going to come there. Next slide we will use. Okay, but there is a problem. Just a second, children. What shall be done? What shall be done? What shall be done? Okay, just give me a second. Yeah, good. So over here, this was the F1 generation. Wait, where it was yellow and white, and here it was yellow and white, and one was brown and red, right? This was the female and this was the male, right? You made the Punnett square, it appeared to be like this, okay, where either this chromosome can come. If this chromosome is coming, it's W, small y, small w. If this chromosome is coming, children, it will be Y plus and W plus, that is brown and red. This is yellow and white. Similarly over here, it can be either this chromosome that is coming, so it will be yellow and white, either this can come, which is basically the plane which does not have the gametes. Now when this and this fuses, you see, you get this. When this and this fuses, you get this. When this and this fuses, you get, okay, this is the chromosome that was there. This and this fuses, you get, okay. Now if you see the traits, if I read down the traits, okay, this trait is yellow white, okay, this trait is brown red, this trait is again yellow white, and this trait is again brown red. So one thing that I saw over here is that I got yellow white and brown red similar to my parents that I started with, yellow white and brown red. So the parental combinations were more over here. Now what about recombination? If I want to try recombination over here, let's try recombination then. So over here, Mendel saw that, okay, sorry, not Mendel, okay, Morgan, while he was studying, he saw that suppose there is small y, there is capital Y, but now I change it over here. Now this Y plus is going this side, and this not Y plus, W plus is going this side, whereas this W is going this side. So over here I will be having red this side, and this red stands for W plus, and over here I will be having green, and this stands for what, children? Normal W, which was not there. This was not the combination. Here we had both the plus plus ones, and here we had the normal ones. But this time recombination has happened. Suppose over here, what was the thing? It was Y and W. There is no chance of recombination happening over here. Now what is the type of gametes that you can get? Let's see. A gamete can have this, that is this, or it can have this, that means Y plus and W, okay. Now in the male case, it can be this, okay, or it can be normal Y chromosome. Now when you fuse, what do you get? YY and Y, W plus W, Y plus Y, and wait, did I mess anything up? Yes, okay. Now over here, what do you get? And over here, what do you get? So basically, if I read down now, what do I get? Y stands for yellow, so I got yellow, but W plus is there, that means yellow red is there now. Similarly over here, this is what? Brown, and small w means what, children? White. Okay, over here what did I get? Here it is yellow, and then again red, and over here it is brown and white. Do you see that here it was yellow, white, brown, red, but here it is yellow, red. This is not a parental combination, this is something new. Over here, this is also something new. These are the gametes that will be formed during recombination. These are the gametes that could be formed during recombination. So you have 25% of chance for this to come, 25% of chance for these gametes to come, or if recombination has happened, then 25% of chance for this to come and 25% of chance for this to come. If recombination was not happening, if the scenario was not there, then 50-50 chances were there for both of these gametes to come from the females. But over here, the chance has been taken away. This is 25, 25, 25 and 25. The female itself is contributing. There is no recombination happening, right? Now, please understand one very important thing that he said after doing all this experiment, that in the case of yellow body and red eye color, or basically yellow body and white eye color, as well as what is it, basically the body color and eye color, he found one thing: the recombination was pretty less. The recombination, the percentage of progenies that were formed like this, it was pretty less. What was the number actually given? Just 1.3%. What was it, children? It was just 1.3%. So only 1.3% recombination was seen, non-parental variety was seen. So what can you conclude with this? Rest 97%, or basically 98%, 98% is basically showing the parental combination. That means probably these genes that you see over here, that is for body color and eye color, they would have been closely linked. They would have been closely linked, which is why they were not showing this much amount of recombination. They just showed 1.3% of recombination. But now if I talk about one more character, if I add one more character, that is the wing size. Okay, that is the wing size. Situation would kind of get different. So you have white miniature version and then one very broad version of the wings. Okay. Now over here he saw that he started off with okay, there is wing size, okay, and he compared wing size and body. Now over here I say the wing size is present somewhere here. The wing size which is there, it is present somewhere at the end. Now don't you think when this and this will segregate, there will be, he started off with white color body with miniature wings. Miniature wings. Now at the end he found that this was changing. Okay, this was changing. You got another wild type of things also. You got just miniature type of things. You got different recombinations that was probably possible. This was not the thing that he got at the end. Majorly there were different types, non-parental types of genes that could be observed in his experiment. With that knowledge, he came to a conclusion that probably the wing size and the body color genes are located at a distance, which is why they showed a recombination of 37.2. I think 37.2, yes. 37.2%. So I'm not making that again. Just for your understanding, I'm saying both of them were close, body color and the eye color. Body color and the eye color genes were close, which is why they showed a linkage of just 1.3%. This is very much important. You have to remember the data. Whereas when he crossed the white color ones and the body color and the wing size ones, he found that 37.2% recombinant progenies were also found. That means 37.2% recombination was happening, probably because they were loosely linked. Body color and wing size was what, children? Loosely linked. On the other hand, if you talk about them, they are closely or tightly linked. So please remember 37.2 for this, okay, 37.2 for this, but just 1.3% for this. That is what you mean by linkage and recombination. Now, I have a question for you guys. If suppose I say that A, B, okay, this is for skin color and this is for hair color. The situation is like this, they are really close. Recombination will be more or it will be less? Tell me, will the recombination will be more or less? It will be less, children, because it is what? Tightly linked. But on the other hand, if you see one gene over here with C, recombination will be there more or less between this and this. It will be more. Okay, please understand. So tightly linked genes would be having less recombination and loosely linked genes will be having more recombination. Yeah. And now one more thing. If there is a gene like this and if there is another gene like this, basically I count it as there's 1 centimeter distance, or basically I say 1% recombination has happened. Basically that means I'm talking about 1 centimorgan of distance. Okay, the one that has 1 centimorgan of distance, will they show recombination? Will they show more recombination? No, they will be just showing little amount of recombination, 1% recombination probably. Okay, that is the thing. Now if they have a distance like 6 centimorgan, then obviously the recombination would be more children. So distance in the case of genes, they are mapped with the help of centimorgan, and 1 centimeter is basically 1 centimorgan. You can think like that. Okay, so please remember the unit. I hope it helps. Now, there is one thing that I told that I will teach you, and that is the comparison chart. So over here they have given a chart and they have asked you tell me which is gene and which is chromosome. So let's try to find it. So occurs in pairs. Both the point are same. I cannot understand which is a gene, which is a chromosome. Next thing, segregate at the time of gamete formation such that only one of each pair is transmitted to a gamete. So basically, according to law of segregation, the chromosomes they segregate, but according to chromosomal theory of inheritance, the factors also segregate because the factors are present on the chromosome. So basically, segregate at the time of gamete formation, it's true for both genes as well as what the chromosomes also. So over here also it is written the same. So again, it's the same point. But over here comes a difference. Independent pairs segregate independently of each other, whereas one pair segregate independently of another pair. Yes, if we talk about two chromosomes in general, okay, this pair will segregate independently of this pair. It can go with this or it can go with this. Okay, one pair segregates independently of another pair. But now when you talk about this, independent pairs segregate independently of each other. That means if they were closely linked, that is basically they are dependent on each other, then they would not segregate independently. They would not segregate independently because they are closely linked. But on the other hand, if they are independent, as in they are distant, these are independent pairs, they segregate independently of each other. They can show recombination and they will be segregating independently. It is not always true that the green one will be always going with the yellow one because they are in distance. So independent pairs segregate independently of each other, whereas dependent pairs would not segregate independently because they are close to each other. They show high amount of linkage. So this is the crucial point by which we can determine that this thing that you are talking about is a gene, whereas this thing that you are talking about is a chromosome. Okay, done. Now we can try to solve the questions. So children, please remember 1865 he published his things, Mendel. 1900 it was rediscovered by De Vries and Correns and Van Schermach. Okay, and then what happened? In the year 1902, Sutton and Boveri started working on this, and they found that when chromosomes segregate, the pair of factors were also segregating. Okay, this was given by Sutton and Boveri. This is known as chromosomal theory of inheritance. Then after Sutton and Boveri did this thing, basically another guy came in, that is Morgan, who wanted to experimentally verify these data. So while he was doing this and working on Drosophila melanogaster, that is the fruit fly, he understood one thing, that there is something that is known as linkage and recombination, which is the exact exception of independent assortment. So according to independent assortments, the chromosomes and the factors, basically there are different genes on different chromosomes and they are segregating independently. But it's not like that. There will be genes, there will be chromosomes that will be having the genes on the similar chromosome itself. At this point of time, they cannot always assort independently. Sometimes they will be dependent if they are showing high amount of linkage, whereas on the other time they won't be. So that is one very important thing. Okay, and then his student was Alfred. Alfred was working on gene mapping. Okay, his student, whose student? Morgan's student was Alfred, and he was working on gene mapping. He was the person who was saying that okay, this much amount of distance is there, then probably this much amount of linkage or this less amount of linkage would be there, and more amount of recombination would be there. Okay, now tell me the answers for the same. Okay, we are discussing it here itself. If Mendel had studied the 7 traits using a plant with 12 chromosomes instead of 14, in what ways would his interpretation would have been different? So according to him, there were, okay, according to him, there were 14 chromosomes, children, and each, we say 7 contrasting pairs of trait he was studying, and each of them are located on different chromosomes. Okay, now he is saying instead of 14 chromosome, if it was 12 chromosome, that means probably the seven of them cannot be 7 of them could not be accommodated, right? So obviously 1 chromosome or 2 chromosome might have more than 1 gene. Okay, these 7 would not be divided into 7 chromosomes, but now would have to divide themselves into what, children? 6 chromosomes. So obviously 1 gene would be extra somewhere. Agree? Now it's asking what would have been, what ways interpretation would have been different if this was the case. He would not have discovered the law of independent assortment. He would have discovered sex linkage. He would have mapped the chromosome. He would have discovered blending or incomplete dominance. Children, he would not have discovered the law of independent assortment, because according to him, one pair, that is in the 7 pairs, one pair is assorting independently of another pair. Now if both the genes are on the same chromosome, do you think independent assortment can be found? Both the characters will be coming. Are the characters independently assorting themselves? No. So he would be failing in here. Okay, he would not have discovered the law of independent assortment. Okay, going ahead, crossing over and diploid organism is responsible. I told the chromosomes, when they cross over, they are responsible for what, children? Tell me. This is second question. Crossing over and diploid organism is responsible for segregation of alleles, recombination of linked alleles, dominance of genes, linkage between genes. Crossing over will be responsible for recombination of linked alleles. It can be more linked or it can be less linked. Anyways, recombination could be found. Okay. So crossing over results in recombination. Going ahead, this is the third question. Experimental verification of chromosomal theory of inheritance was done by Mendel, Sutton, Boveri or Morgan. I told you there is only one guy for experimenting things, and it was Morgan. Okay, this came in NEET 2020. Select the correct statement from the ones given below with respect to dihybrid cross. Okay, let's read it very carefully. Okay, tightly linked genes on the same chromosome show higher recombination. They are tightly linked. That is the first situation. A and B, and it's saying that since they are tightly linked, they are showing higher recombination. Is it true or false? Can tightly linked genes do more fight? No, children. This is wrong. Genes far apart on the same chromosome show very few recombination. It's saying it shows less recombination. So more if you are not friends with your friend, so basically you have less fight. Is that true? Again, it's wrong. If you're close, then only. Now genes loosely linked on the same chromosome show similar recombination. What is similar recombination? We cannot define that. Tightly linked genes on the same chromosome show very few recombination. Tightly linked genes on the same chromosome shows very few recombination, and this is exactly true, children. This came in 2010, and this will be your fourth question if I am right. Okay, done. Going ahead, what do we have? Which of the following statement is not true of 2 genes that show 50% recombination frequency? Now children, please remember, 50% is the maximum recombination possible. 50% is the maximum recombination possible. Okay, that is the maximum recombination possible. There is no 100% of recombination. Now there are chances that other 50 is for normal parental type, other 50 is for the recombinant type. So only 50% is there, please remember that. Now which of the following statement is not true of 2 genes that show 50% recombination? So it's the most highest one. So these genes show independent assortment. If they were showing independent assortment, then there is no point of recombination, right? They are independently assorting, there will be 2 characters. If the genes are present on the same chromosome, they undergo more than one crossover in every meiosis. The genes may be on different chromosomes. The genes are tightly linked. They are asking you what is not true of the genes that show 50%. So this is true. Okay, this is true. Okay, I messed it up. Okay, just a second. Okay, if they are independently assorting, that is something different. Obviously they will be showing 50% recombination frequency, because anything could happen. The linkage, they are not linked at all. They are not linked at all. So obviously there is one chromosome like this, there are another chromosome like this, they are independently assorting, there is no linkage. So obviously recombination can be possible in any ways. So that is true. If genes are present on the same chromosome, they undergo more than crossovers in every meiosis. Yes, if genes are present on the same chromosome, they undergo more than one crossovers. Yes, that is also true. Now genes may be on different chromosomes. Yes, it can be on different chromosomes. Then also there can be 50% recombination. Okay, because they are not linked, so obviously recombination is possible. Now it can be like this, different, or it can be on the different chromosome as well. The recombination would be more either way. Okay, but if they are close, the genes, when they are tightly linked, can they show 50% of recombination? That is not possible. So this is the correct answer. We had to find out which one is not true. For the ones that show 50% recombination, this is not true. Okay, I hope this is clear to you. Got it? Now going ahead, we solved all these questions, children. Basically this was the answer for this. Crossing over, recombination, Morgan was the one, and tightly linked genes on the show very less combination, the genes are tightly. These were the answers. I explained it to you right now, so I am not explaining it to you again. See for yourself if you got the answers correctly or not. Now there is one little question for my little champions over here. Okay. Okay, a human male produces sperms with genotypes this. Okay, now you have to tell what is the corresponding genotype of this particular person. So you have the genotypes like this. What is the corresponding genotype of the person who is producing these gametes? Can you tell that? That is something out from, I think we did it, right? And I think we did it. I'm so sorry. I'm so sorry. Wait. Okay. For today's homework, I can give you a question right away. Just a second, just give me a small second. So over here, I have written a question for you. Please try to solve this question. This came in 2002, children. Okay. Now the question is, there are 3 genes A, B and C. Percentage of crossing over between A and B is 20%, is just 20%, okay. B and C is 28%. And A and C is 8%. A and C is 8%. What is the sequence of genes on the chromosome? BAC or ABC or ACB or none of this? Can you try solving this? This will be the homework. This we did previously already. Okay. So can you try doing this? And do let me know in the comment section A, B, C or D what is the answer. Okay, whatever is the answer, you can tell me in the comment section itself. So that's all, children. Now, if you think that this particular video was helpful for you, you understood linkage properly, then do subscribe to the channel and also comment down. I'll be reading down your comments. And also, if you have doubt, children, please do not hesitate for even 1 second. You can just ask me your doubt. Okay, I'm here to help you only. So bye-bye, children. Take care.